This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only.
Good afternoon. My name is Pascal Gagneux. I'm the Associate Director of CARTA, and it is my pleasure to welcome you all today for this public CARTA Symposium on early hominids. We're very fortunate to have a cast of eminent researchers from 4 different continents, and I'd like to thank all the speakers for traveling very far, in some cases, to make this conference possible. And with no further ado, it's my pleasure to introduce Dr. Fred Gage from the Salk Institute, CARTA co-director.
Thank you, Pascal, and I'd like to add my welcome to you all. So CARTA actually is the Center for Academic Research and Training in Anthropogeny. So what's anthropogeny? The most recent definition of the term from the Oxford English Dictionary is investigation of the origin of humans. However, as early as 1839 in the Hopper Medical District, it was defined as the study of the generation of man. So we have a mission statement for this organization, and it is to use all rational and ethical approaches to seek all verifiable facts from all relevant disciplines to explore and explain the origins of the human phenomenon. An important caveat here is while we minimize the complex organizational structures and hierarchies that often infest these kinds of organizations and avoid any unnecessary paperwork and bureaucracy, the hope is to get to the science and get the discussions out front. This would not be possible without our major sponsors who've, with great foresight, sponsored this organization, the graduate program that surrounds it, as well as these conferences. And the first one is the G. Harold and Leah Mathers Charitable Foundation based in New York. And secondarily, but not least, Annette Merrill Smith is very much appreciated for her continued support of this effort. These individuals are foresighted and extremely important for us.
Thank you all for coming, and now it's my wonderful privilege to introduce Michele Brunet from Poitiers.
So, when I began to look for early hominid, the situation was very clear. Early hominid was known in South Africa and in East Africa on its whole. And when I decided to go west of the rift, I was a young boy at this time, 25 years ago. I'm still a young boy. You don't believe? So, okay, we shall speak at the end. Every people say, Brunet, you are crazy. There is no fossil west of the rift. So, we can try. I go to Cameroon first because there is a war in Chad. There is very often a war in Chad. And in 1994, the war stopped, and I go to Chad. It means 16 years ago. And now in Chad, we have 500 localities and more than 20,000 fossils. But it's not the end of a story that I'm telling you today. It's just the beginning, just the beginning. We are very lucky. We are working in a very, very nice desert, Joab Desert. Here, you see? Of course, you need a lot of jeep. Gemini is here, Lake Chad, and we are working here, south of Tibesti. Lake Chad at this time is 5,000 square kilometers. 5,000 years, Lake Chad was 400,000 square kilometers. It was mega lake shade. And with my team, we show that you have a succession of wet and dry period at least during the last 8 million. All the sites you see in green is the Lake Chad Basin, and in black here is the shore of the last mega Lake Chad. The actual Lake Chad, and we are digging in this part. You have here Aeolian, perilacustrine, and lacustrine facies, and it's cyclic. You see our work? Chad is a very nice place for geologists. All is flat. Tectonic is very quiet. It's the only thing which is quiet in Chad. And of course, our geologists are very happy, because if you want to make an outcrops, you have to do it. Nice work, nice job. We have a lot of fossils. This one is a very interesting one. This one is an Anthropogeny, extinct mammal. It's a sister group of hippopotamid, amphibious mammal, big one, big like hippopotamus. Which is very interesting is that with 2 miles in Chad, we have the same species of anthracotherid that you have in Libya in the Sirte Basin. It means that you have the same bioprovince at this time between Libya and Chad. It means that 2 miles can go to Libya or can go from Libya to Chad. And you have no anthracotherid east of the rift. East of the rift, it's another bioprovince. We have fossil with water, fish, croc, birds. We have amphibious fossil, forest, colobin, some cousin of elephant, Dinotherium, in forest too. And we have equid, Hipparion, antelope, lepus, in savanna. It means that we have mosaic landscape. Probably something which is very close to the actual Okavango Delta. So it means that probably Toumai was living in such a landscape, and in this landscape, he was probably living in a forest part. In Chad, we have fossils from 3 million to 7 million. The first hominid we found is an Australopithecine, which is dated around 3.5 million. It is known by a large jaw and some other teeth which has not published at the moment. So, first thing, remember, pre-human are just known South Africa, East Africa. Now, you have Australopithecine in Central Africa. It's a little bit different. Toumai. Toumai is older. We have two datations for Toumai. A biostratigraphic datation by the evolutive degree of the mammalian assemblage with Toumai, which give us something around 7. And we have, with the cosmonucleate beryllium-10, the same datation. Above Toumai, 683, behind 712. It's around 7 million. We have lower jaw, and you can see, just Tim told you about the feminized canine. You have a lower canine here, very small one. The cranium was a little bit crushed. You imagine if you have your cranium doing 7 million in sediment. It's a long time. So we make deconstruction, reconstruction, as you can see on this. And you have here the 3D reconstruction. This one, you have the cast here. You have the virtual image on the screen, and you have the cast here. Okay? I put it here. It's not virtual. You can touch it. Biped or not biped? It's clear. Just like Ardipithecus, the basic cranium is shortened, and the foramen occipital is forward, as you can see on this picture. When you have a cranium like that, you can reconstruct. We do that with an artist in Paris. Why he opened his mouth? Because we are very often discussing together. Why not? We are friends. He's not lucky because I am in San Diego. He's still in Poitiers. So I think that you are looking for the moment at the earliest hominid known phase. And I think that it's probably like that, it means that if Toomai did some bad thing in the university here, if you have this picture, you can catch it. And now, what we know. Look, that I call late Miocene hominid. Late Miocene hominid is just a synonym that of my friend Tim called Ardipithecus. I'm right? Yeah. No, because I checked my English. I don't understand whole. So, late Miocene hominid, what you have? You have two east of the rift, one in Ethiopia, one in Kenya. All are around 6 million. And they are very close in terms of geography. And remember, they belong to the same bioprovince. East of the rift, a little bit older, probably 1 million older, we have another one, Toomai, in Chad. I think that it's just the beginning of something, just. Because, of course, look at this map. Here you have the beginning of a new story, surely. We are still digging. I'm digging in Libya. I'm digging in Egypt. I just come back from Cameroon. So, when you invite me in 20 years, I am still young. I shall have a lot of new things to show you. So, which is very interesting is that the repartition is completely different, as you can see. All these results are published in international peer review magazine, as you can see. It's just a summary. Panid and Hominid, two sister groups. We need more, as Tim said, but for this time, we have some idea about already a lot of things. Thanks. Nice picture, no?
And I'm now going to introduce my colleague, Johannes Haile Selassie. Johannes is from the Cleveland Museum of Natural History. He'll be talking about the earliest hominids from Ethiopia. This is something you don't want to miss. Watch for the name Johannes Haile Selassie. He has new sites, new fossils. He's going to challenge Brene.
Good afternoon. Today I'm going to talk about the earliest hominids from Ethiopia. When I got this title, I was thinking about how do I determine the earliest hominids when we have more than 20 species of early human ancestors. So I'm going to talk about those that were found in Ethiopia only. Now, as I said, there are about 22 early human ancestor species that are known in the fossil record. And when it comes to our evolutionary history, Ethiopia is usually dubbed as the cradle of humankind. Now, why is that? And I'm showing you here a phylogenetic, sort of a phylogenetic tree of the human family tree. And when you look at how many of these taxa have been found from Ethiopia, there are about 11 of them that were found from Ethiopia. So that tells you why Ethiopia is usually dubbed as the cradle of humankind. It has some of the earliest hominids that I'm going to talk about today, and also some of the younger hominids that, as you see from this slide. So the question is, why do we have this many hominid fossils from Ethiopia? And Dr. White explained a little bit about this. Ethiopia is a country located in the Horn of Africa, as you can see here. And geographically, it's situated in an area where 3 different plates of the earth are moving apart from each other and creating this huge rift valley. And this rift valley has been expanding, and it is still expanding because of tectonics. And it has luckily exposed about 6 million years of our evolutionary history. And that's why people have been going to this place for the last 4 decades and looking for some of the remains of our earliest ancestors. When you look at the distribution of these hominids that I mentioned earlier, they're mostly located within the Afar rifts area, as you can see here, and some of them extend to the main Ethiopian rift and also to southern Ethiopia. So here is the secret behind why Ethiopia is really the cradle of humankind. Now, the earliest hominids I would usually call are the ones that are older than 3 million years. And these include Ardipithecus cadaba, Ardipithecusramidus, which Dr. White also mentioned, Australopithecus anamensis from slightly younger deposits, and Australopithecus afarensis, which is the species to which the famous Lucy belongs to. But obviously, I'm going to talk about the earliest 2 early human ancestors, Ardipithecus cadaba and Ardipithecusramidus. Now, the big question is, we know that our family is Hominidae, but we ask ourselves, what kind of species should be included into our family? There has to be specific characters that refer to this family Hominidae. And basically, there are 2 major characters that define the family Hominidae. One of them is being bipedal, walking on 2 legs. We're the only mammals who are habitual bipeds. And the second character has to do with the teeth. Hominids do not have the canine honing function like apes. So we don't hone our upper canines by rubbing it against the lower premolar, which apes do. We have changed our social structure that we didn't need that honing function anymore. So those are the 2 characters that usually define the family Hominidae. So by using this character, you have early human remains from 5.8 million years ago. And when they were found, they were 1.4 million years older than the oldest known at that time, Ardipithecusramidus. So you have to be really careful in terms of recognizing this as belonging to the family Hominidae. So we had to look for the specific characters that enable us to include them into the family Hominidae. So this is what we had to look for. But obviously, Dr. White showed you that the earliest hominids fossils could actually be fit into a shoebox, which is true. Most of the specimens that we had when we named Ardipithecus cadaba in 2001 were 17 specimens, mostly isolated teeth. But of course, based on these remains, we tried to look at what characters distinguish this early hominid from the other known taxa of early hominids so that we don't make mistakes in terms of recognizing a new species when it actually belongs to something that was already named. So we had to look for specific characters in this early hominid species. It's very limited in terms of its geographic distribution. Most of them come from the western margin of the Middle Ash study area. This is the rift margin, the western rift margin of the Afar rift. And we found most of the specimens right along the rift edge right here. But we also had 1 specimen from slightly younger deposits at 5.2 million years from Amba East. These are between 5.5 and 5.8. That was the youngest specimen we had actually included into the Ardipithecus cadaba hypodyme at that time. But we also have some Ardipithecus cadaba remains from farther north at a place called Gona, which my colleague Dr. Semaw will talk about a little later. So as you can see, what we had was mostly isolated teeth with some postcranial elements, as you can see here, some arm bones, including the toe bone that I mentioned from the younger deposit. So based on this, what we tried to do was to look at the major characters that we could pick up from these dental and postcranial elements and see if it actually belongs to the family Hominidae. But of course, we're talking about the earliest hominid, and obviously it's going to have a lot of primitive characters that are probably closer to the common ancestor that we shared with chimpanzees. So the one thing we looked at, because we had an upper canine of Ardipithecus cadapa, was the morphology of this tooth, because it has a honing function in the apes, and we wanted to look at whether it had honing function or not. But the first thing is we see that this canine is really primitive. And also the lower third premolar looks very primitive. So that's something that distinguishes it from the known earliest hominid at that time, which is Ardipithecusramidus at 4.4. We also tried to look at, see if Ardipithecus cadapa had a honing function, as in chimpanzees. This is a female chimpanzee that you see here, and this is a composite of Ardipithecus cadapa teeth. And what we realized is that it is really primitive, but not like an ape. It was not really, we can't really say for sure if it was honing its upper canine by rubbing it against the lower P3. But interestingly enough, the lower P3 had a little facet on the side, which indicates that it was actually overlapping with the upper canine. But we can't really be sure whether it was there was a functional honing mechanism in Ardipithecus cadapa or not, because the lower canines tell us a different story in terms of their morphology. So obviously that could be simply a relic of what they got from the common ancestor we shared with chimpanzees. The other element from the younger deposits that we included into Ardipithecus cadapa was this toe bone. And this is the toe bone that we used to infer bipedality in Ardipithecus cadapa. Now, after we looked at Ardipithecusramidus, particularly Ardi, we had to reconsider our conclusions from the first one, and now what we're saying is that habitual bipedality, since we didn't even see it in Ardipithecusramidus, remains to be ascertained by further discoveries, and we're still looking for more discoveries in this part of the Middle Awash. So that's about what I have to say about Ardipithecus cadapa. The next oldest is Ardipithecusramidus. Now, Ardipithecusramidus is a species that's been known from the deposits, about 4.4 million year old deposits from the Middle Awash. And it's also known from further to the north from a site called Gona. And I'm sure Dr. Semaw will mention that. But this is where Ardipithecus was initially found. The first specimen, as Dr. White mentioned, was found in 1992. But in 1993, there was another important specimen found from Aramis, which is this child's jaw that you see here, which had the milk molar. Now, this milk molar had to say a lot about its morphology and which way it's related to. Obviously, it's not like entirely ape-like, but it's not also like hominid-like, like later hominids. So it was intermediate between apes and hominids, and it shows how primitive it is, and it warranted its distinction from other species of Australopithecus. So the first piece of RD, which is the most critical specimen in the Ardipithecus hypodyme, was found, the first piece was found in 1994, 3 years excavation, 17 years of curation, preparation, and analysis resulted in the 11 paper publications in Science that Dr. White mentioned earlier. But it's very interesting because we learned a lot from this partial skeleton in terms of its morphological features. Now, I want to mention some of the highlight points of RD in relation to what we know of the later hominids. Now, when you look at the cranium of RD, which I'm sure Dr. Asfa will talk more about later, it had small brain. Its face was not as projecting as in chimpanzees, and probably comparable to Tumai, which Dr. Brunet showed us, and which is right here. In other aspects, the canine of RD is not like a chimpanzee. It's large. It had large canines, but it's not like in chimpanzees. The crowns of their teeth were shorter, and there was no sexual dimorphism between males and females as much as we see in chimpanzees. So those are like derived characters that put them into the family Hominidae. And moreover, we don't see any functional CP3 honing complex. Ardipithecus was not honing its upper canine like apes do. So that differentiates it from apes and puts it into the hominid family. So based on what we learned from its teeth and the overall morphology and size, it was not a specialized, it didn't have a specialized diet. It was more of a general omnivore and frugivore. So this is what we learned from Ardi's teeth. We also had, luckily, complete hands of Ardi. And what we learned from the hand elements is that it had a very flexible palm. It was not, or wrist, it was not as rigid as the knuckle walker cousins like chimpanzees and gorillas. And we don't also see any characters of suspension or vertical climbing in these hands. So it was more adapted to palmigrade clambering, and we don't see any sign of knuckle walking in this hand. So these are very important specimens that as you will see later. The other element, which is very critical in terms of inferring its locomotion, is the hip bone. Now, you saw a slide from Dr. White's talk of the pelvis, which was crushed and it had to be reconstructed, but there were very critical features on the pelvis that were not distorted, which actually tells us about its locomotory behavior. Now, as you know, the pelvis or the hip bone is not just an obstetric organ, it's also a locomotory organ. So it tells us about how this animal, how animals move about by looking at the morphology. So here we have a comparison of 4 pelvis. This is a modern human. This is Lucy's pelvis. This is Ardi, and here is a chimpanzee pelvis. Now there are major characters that distinguish it from the apes and that are human-like include a feature called anterior inferior iliac spine, which is present in bipedal animals and not in quadrupeds. Another feature, which is called the greater sciatic notch, which is also present in bipeds and not quadrupeds. So these 2 characters seem to be shared by humans, afferences, or Lucy, and Ardipithecus, some indications of its bipedality. But at the same time, it had a feature that's more like an ape, which is the position of what we call the ischial tuberosity, which is more like an ape than it is like the humans. So she had a mosaic of features that sample derived hominids and also apes. But you look at the foot, I don't have to get into this in detail because Dr. White has already mentioned about it, but it's really interesting that it had an opposable toe. It's really ape-like foot, and it had curved phalanges, the fingers were really curved, and the grasping big toe, and it didn't have any arch. Obviously, she wouldn't have been part of the army with that foot. So we have this primitive ape-like characters. So what is she? Obviously, there is evidence that she was able to walk on 2 legs on the ground, but not in the way that we do it today. So she was a facultative biped who was able to walk on 2 legs on the ground, but also able to climb up on trees. But because of her and what we know from her, a number of hypotheses that existed before were tested. And let me tell you some of these hypotheses. One of them is what's called the savanna hypothesis for bipedality. The other one is that we evolved from a knuckle walker ancestor that we shared with chimpanzees. And another one has to do with chimpanzees being good models to reconstruct the common ancestor we shared with them. Now, what we learned from Ardi is that she was not living in open savanna environment. She was living in a more woodland, forested habitat. So that savanna hypothesis was falsified. Of course, that was falsified with other evidence too. Did we evolve from a knuckle walker? No, she doesn't show any sign of knuckle walking on her hands. that gets falsified. And the other one is, obviously, chimpanzees cannot be good models for the common ancestor we shared with them. Because think about this, chimpanzees have been evolving so much since they split from the common ancestor they shared with us. It's really interesting to see that our distant cousin, gorilla, in some cases share character with humans than chimpanzees do. So that tells you how much chimpanzees have evolved. So using chimpanzees as model is really wrong. So that was also falsified. So in general, when you look at what's come up since Ardi, it looks like it causes a major paradigm shift in terms of how and where we should look for the model for the common ancestor that we shared with chimpanzees. And not only that, it also cautions as to how we define the family itself, the family Hominidae. Are the two characters that I showed you earlier enough to define the family Hominidae? Maybe not. So when it was published, it was recognized as a major scientific breakthrough, at least in human evolutionary studies, and that is really true. So that's what I have about Ardi. But slightly younger earliest hominids would include species like Australopithecus anamensis, which is also known from Ethiopia, largely known from Kenya, from deposits dated to between 3.9 and 4.2 million years ago. But we also have anamensis remains from the Middle Awash, from localities where Ardipithecusramidus was found. And our Australopithecus anamensis in the Middle Awash was found from slightly younger horizons at same localities. So that might strengthen the hypothesis of Ardipithecusramidus being the ancestor of Australopithecus anamensis, which is more likely as we know it right now. The other one, the other species, younger, but still earliest hominid, is the species which includes the famous Lucy, Australopithecus afarensis. Now, this is one of the species, early hominid species, that is well known because we've known Australopithecus afarensis since the 1970s. And there are hundreds of fossil remains of Australopithecus afarensis, mostly from Ethiopia, but also known from Kenya and Tanzania. So we have a wealth of information about Australopithecus afarensis than any of the other taxa, early hominid taxa. Now, partial skeletons are really rare in the fossil record. There are probably 5 or 6 partial skeletons in the entire human evolutionary fossil record. And it happens that we have most of them belonging to the species Australopithecus afarensis. Lucy is the first one, which was found in 1974. And then in 2003, an Ethiopian paleoanthropologist, Ellison, announced the discovery of a partial skeleton of a child of Australopithecus afarensis, which he nicknamed Salam. And recently, as Dr. White mentioned, we announced another partial skeleton of the same species, nicknamed Kadanumu. And this partial skeleton is dated to about 3.6 million years old. And it's about 400,000 years older than Lucy. Now, what's really interesting is that we still have gaps in the fossil record, as Dr. White mentioned. And what Kadanumu and all the other specimens that we're finding from this new site called Oranzomille is that they're filling some of these gaps that exist between Australopithecus anamensis and Lucy's species Australopithecus afarensis. So what that means is that we can actually see how these two taxa are related to each other by filling the gap in the fossil record right here. Right now, a lot of people agree that Australopithecus anamensis is the direct ancestor of Australopithecus afarensis, or Lucy species. And that hypothesis would be better tested if we could actually fill this gap in the fossil record right here. But what we're finding here already shows that the hypothesis of ancestor-descendant relationship between these two taxa cannot be falsified. But it challenges the idea of actually recognizing two different taxonomic names, probably for a single phyletically evolving lineage. So the suggestion right now, at least as far as I'm concerned, is that Australopithecus anamensis and Australopithecus afarensis are two ends of the same phyletically evolving lineage. And I'm suggesting that we should probably use just afferences to include everything from 4.2 to 3 million years. But of course, some people have issues with that. But that's what I have, and I could talk about Australopithecus afarensis and anamensis for probably another hour, but that will be a subject of another lecture, and I'll stop here. Thank you.
Thank you very much, Dr. Haile Selassie, for a wonderful presentation, bringing us back to anatomy and letting all of you students know that if you want to study human evolution, you're going to have to learn your dental and postcranial anatomy. Last year, many of you know, was the 200th anniversary of Darwin's birthday. And wouldn't it be amazing if Darwin were here today to see the amazing evidence that people like Johannes Haile Selassie have been finding in the Ethiopian desert to show how we evolved. This evidence is overwhelming at this point. We're learning more and more as the gaps are closing. And one of the gaps in the geography of this part of Ethiopia is between the Lucy site and the Ardi site. And you might be wondering, what's going on there? Well, you're going to find out now from Dr. Sileshi Semaw of Indiana University, who runs a project known as the GONA Project. You know about the Lucy Project and the Ardi Project. You probably haven't heard as much about the GONA Project. Sileshi is the only archaeologist in today's symposium, but he's been forbidden to talk, at least talk very much, about archaeology, because stone tools, material culture, don't start until about 2.7 million years ago. And Dr. Semaw is the scientist who demonstrated that and found the world's oldest stone tools. It turns out, though, that those stone tools are about halfway through a sequence in the GONA study area that goes from quite young deposits to very, very old deposits of the same age that Johannes was speaking. So Sileshi is going to take us through that wonderful new succession. This is really exciting research, and we're all privileged to have him here. Dr. Sileshi Semaw.
Thank you very much, team, for the clear introduction. All my colleagues have seen me always talk about the oldest stone tools from GONA, which I really love to talk about, and I'm not going to talk about those today. Today, I would like to talk about the earliest hominids, which is the theme of the conference here. Professor Michel Brunet did mention two sites in East Africa that are late Miocene. There aren't only two. GONA has also late Miocene hominids, and it's a third site in East Africa with late Miocene hominids. The oldest stone tools were found prior to 1999. And in 1999, the GONA project started as a large multidisciplinary study, and we moved from the younger deposits to the older deposits, and we did find two major hominid discoveries in the older deposits in the late Miocene, dated to more than 5.5 million years ago, and Ardipithecusramidus, originally found in the Middle Awash study area, and then also at GONA in deposits dated between 4.5 to 4.3 million years ago. The GONA study area is located north of the Middle Awash study area, where a large number of Ardipithecusramidus fossils and also cadaver were found. West of the famous site of Lucy, Hadar, and south of Waranso Mille, where Dr. Johannes Haile Selassie works. This is the Gona Paleoanthropology study area. On the oldest stone tool localities are right here on the eastern part of the study area. After 1999, after I finished my dissertation work with the oldest stone tools, we started surveying west and southwest. The deposits down here are much younger. They go to the later part of the Pleistocene. As you go up here and west, the deposits have been uplifted, and we have 4.5 million year old soil formations about here, where we have Ardipithecusramidus, and then as we move further west, we have even older deposits going back to 6 million years ago, where we have just a few dentition attributed to Ardipithecus cadaver. The GWM labels are for the Gona western margin sites where we have Ardipithecusramidus 4.5 to 4.3 million years. And ESC means escarpment for the Ardipithecus cadaver sites, for the older sites. Just to give you a feel of what these sites look like, I have a red arrow to show there are a bunch of people doing survey in there. It's a very huge area, but unfortunately, I'm sure it is the same in the Middle Awash study area where they survey and search for Ardipithecus cadaver. This area is covered with vegetation. So as you can see here, just a close-up. So you'll be lucky to find exposures that are fossiliferous, where you can find hominids and fauna. Some areas like this one, just a little bit of sparse vegetation, but a lot of fauna. You can see project members doing survey. The area is covered also with gravels and cobbles and fossils. They don't have really much time to survive in this kind of situation. So to be there right time will be really lucky. This small spot is just one of the isolated tears attributed to Ardipithecus cadaver found in this area. And when we find fossils, we collect all the gravels and cobbles. We crawl shoulder to shoulder and pick every fossil bone to be later investigated to make sure that we are not missing any fossil bones, because as you go further back in time, going to deposits that are older than 5 million years ago, pretty much what you find are just dentition, as shown by Dr. Haile Selassie. There are just bits and pieces of the postcrania, the fossils below the neck, found from this time period. And in those regards, Professor Michel Brunet is lucky to find Tumai in deposits that are close to 7 million years ago. And all combined, these are the fossil dentition that we found from Gona, which are dated somewhere between 5.8 to 5.2 million years ago. Conservatively, I would say about 5.5 million years. Just the first specimens from the Middle Awash, which Dr. Haile Selassie just talked about, I just put them here for comparison. We have more or less the same specimens in terms of dentition. How do we know how old they are? There are soil formations right above, stratigraphically above the areas where we found these tears. And one of the areas called Balewa, we have a complex of tophaceous exposures in that area dated to about 5.4 million years. So that's a minimum age for the fossils we have from the Gona study area. And another site called Bordelle, where we have fossil teeth attributed to Kadaba, a tuffaceous exposure found below this Sifi tuff is dated to 6.5 million years. We don't have fossils that go all the way to 6.5, but the oldest we believe could be somewhere around 5.7, 5.6, but conservatively I would say 5.5 million years. This is a Bordelle close-up of the tuff that was dated to 6.48, and even closer look down here. And we have more than half a dozen volcanic ashes that were dated in the entire sequence, going from something dated about 5.2 all the way down to 6.48 from Bordelle. All right. This is about it for the late Miocene discoveries that we made at Gona. The western margin from the Middle Awash also extends north into the Gona study area, and we also have fossil hominids assigned to Ardipithecusramidus at Gona. And this is the first area we did survey in 1999, and just the first day from camp, it took us almost half a day to drive to this area, and you are going up. The deposits as you go up, because of the uplifting, are much older than where we do archaeology, which is my specialty. So the first day we get there, we found this mandible, and it was found by the great fossil finder at Gona, an Afar named Asa Ahmed. And we continued in 2000, 2001, and we started finding more, usually dentition. And there is a lot of work involved, sieving and building dam to keep the fossils from eroding and getting lost, when we come next year. And all in all, by about 2004, 2005, we had these fossils, including the one we found in 1999, and also phalanges toe bones, which show bipedality, also from Gona. And the dating below where we found Ardipithecusramidus fossils at Gona, we have a fossil, a volcanic ash dated to 4.47, a maximum age for the Gona Ardipithecusramidus, about 4.5. It's a close-up of the dated tuff right here. We don't have good minimum age, but based on paleomagnetic profiles, the minimum age for the Gona Ardipithecusramidus is 4.3. We have two volcanic ashes dated to 4.5, close to 4.5. And so the Gona Ardipithecusramidus are dated between 4.3 to 4.5 million years. And Gona is the only other site that has both Ardipithecusramidus Kadaba and Ardipithecusramidus ramidus. And this was published in 2005 in Nature. And we did continue after 2005. This is one very important Ardipithecusramidus site. We did find a complete upper jaw from this area, which is awaiting publication. Again, it was found by Asa Ahmed. This is Busidima River, and the site is located just behind the hill here. Just early in the morning, 8:30, he found this molar. He showed it to me. We stopped the car, we walked in there. And this is the site. We found a lot of associated molars, which we will be publishing very soon. So in conclusion, outside of the Middle Awash, Gona is the only other site where we have the earliest hominids, including Ardipithecus cadaba, about 5.5 million years or older, and Ardipithecus ramidus between 4.5, 4.3 million years ago. And I will be talking a little bit about the oldest stone tools in the public talk tomorrow, not today. Thank you very much.
Thank you very much, Dr. Sileshi. For those of us who've been working in this field for some time, this is illuminating a period of time that we had no data from. It was a complete black hole until just 10, 15 years ago. And it's due to the hard work of people like Sileshi and his crew that we're really gaining knowledge into the biology of what these hominids were like before 3.5 million years ago. We can learn a lot more from them if we squeeze them hard, so please come back where you'll learn about their teeth and their heads and their hands.